Good morning or afternoon, everyone. Welcome to our live demo webinar focusing on how to automate 96 throughput NGS library prep with the OpenTron's Flex high throughput NGS workstation. My name is Nick Ritzo. I'm a product manager for hardware and BD OEM here at OpenTrons, and I'll be joined today by my colleague Matt Akana, a senior NGS scientist at OpenTrons. Today, we're going to walk through automating your NGS workflow and more specifically, how the OpenTrons Flex high throughput NGS workstation sets you up for success with this application, including hardware features that we've already debuted, such as our 96 channel pipette, the OpenTrons on-deck thermal cycler, magnetic module, and then we're also going to be featuring the new Flex Stacker module, which allows you to increase your tip and plate capacity. And then we're going to run through a short demo of the NGS workstation in action. And we'll also present some data out from the science team that we generated automating some common NGS library prep chemistries. Before we do get fully kicked off here, I'm going to pause for just a moment, make sure everyone who signed up can join us. And I'll also use this time to plug our demo room, which is where we are now. We are in our headquarters, which is in Long Island City, New York. If you or anyone on your team are based out of or visiting or even just close by and want to come visit New York City or the tri-state area, please feel free to work with your main point of contact at OpenTrons to schedule a time to come and visit us and see the OpenTrons Flex and all of these amazing modules in person. It's a really cool space, and we would love to share it with you. Okay. So just quickly, looks like we are good to go. And just a quick reminder to everyone that this webinar is being recorded and will be made available on the OpenTrons website. We will also be sending a copy of the recording via email to all who registered after the webinar. To help with the overall webinar experience, all attendees are on mute. If you have a question, please use the Q&A box on your screen, and we'll answer your questions at the end. We'll have a little bit of a Q&A session then. Okay, first, just to kick us off, I'll say a couple of things about OpenTron's ethos and why we exist. It's our mission here to support scientists doing groundbreaking, valuable research with powerful, accessible lab automation that frees you up to do more than just pipette. We've helped thousands of labs all across the world automate an extensive workflow list. The OpenTrons Flex is our newest generation robot, and we're excited to show you how it can help you and your lab increase your throughput and automate your NGS workflows with both existent modules and what we are debuting. First, I would also like to kind of just chat through some key points to consider when you're thinking about how to automate your NGS library prep. Let's talk about the technique and chemistry. Not all NGS library prep is the same when it comes to automation. Some techniques require more pipetting or smaller volumes or different modules on deck. It's important to select kits that are more automation friendly. These kits will typically use magnetic beads instead of spin columns for cleanup and have a higher dead volume to allow for proper pipetting without needing to tilt and worry about dead volumes quite as much. Another adjustment that you can make is to use enzymatic normalization to normalize your libraries rather than qPCR-based methods. These are more easily automated and are very quick. If you want any advice on automating your chemistry, please feel free to reach out and talk to us. Transitioning from a manual process to an automated process is not always a one-to-one technique translation, but ultimately will yield faster, better, and more consistent results for you and your lab. And we are here to help along every step of that journey. We've automated hundreds of different NGS protocols in our and our customer labs, and our FAS team also has kind of run along many of those studies to show you what each step of your automated protocol could and should look like. Next, let's discuss throughput. the smallest automation systems typically support 24 well sample throughput, with larger systems supporting 3 to 4 sample throughput or more. Of course, those larger samples are a larger investment, typically several hundreds of thousands of dollars. The Flex supports up to 96 sample throughput in a benchtop format at an unmatched price point. We do have the ability to scale up and utilize 384 well plates on the Flex, but the highest throughput head that we currently support is in the 96 sample format. And finally, kind of a consideration around lab and bench space. Question I hear all the time from folks is, how much space do we actually need to free up in our lab to have space for automation? Many automation systems that can support NGS are large. This is often an issue with folks who are trying to get this into their space where lab and bench space is so valuable and comes at such a high price, and really is a consideration for how the flow of your entire lab works. The Flex is about a 3-foot cube and only takes up about 2/3 of a lab bench. This is about as much space, this is about as space efficient as it gets for up to 96 sample throughput. And another key concept that we'll be talking about today is how you can increase that throughput with the stackers while minimally increasing that bench space kind of footprint. And also, as we go through and talk about the modules that sit indented into the Flex deck, you can also see how, because those are all existing within that kind of 3-foot cube, why it means that with other solutions that need to have those outside or below or next to the robot, why that takes up so much more room. So now I'm going to just go over a few key features of the Flex high throughput NGS workstation, and also run a short demo so you can see it in action. We'll take all questions at the end, but again, please feel free to type them into the Q&A box at any time, and we'll ensure that we get to all of them at the end. If there's any questions that we don't have time to answer, if you are registered with your email, we can always reach back out to you afterwards and kind of track that information down. And as a last resource, always feel free to reach out to your OpenTron's local rep. They are also a wealth of knowledge for you. So next to me here is the Flex open high throughput NGS workstation. The Flex has kind of several key features here. First is going to be the gantry. Here, this has the ability to hold several pipettes. On this currently is the 96 channel. This currently only comes in one size, which covers 5 to 1,000 microliters. However, the gantry can also support our single or 8-channel pipettes, which come in 2 size versions, a 1 to 50 or a 5 to 1,000, and it can support any configuration of those. So it can have 2 single channels or 2 8-channels. But for the high throughput kind of workstations here, we will really recommend that you use the 96 channel. The 96 channel does also have some unique features, such as partial tip pickups. So it can perform similar functions, such as the single channel, where it has the ability to pick up 1 pipette tip or 8 pipette tips at a time, therefore kind of freeing up what you are able to do, hit picking, things like that, with this singular head that you can use on the robot. Also mounted to the gantry over to the right-hand side, which will be kind of shown as we go through this protocol better, is the gripper. The gripper does not block any of the pipetting ports, so you can have this with any pipette configuration, and it has the ability to move SBS format plates around the deck. In this protocol, you will see it kind of move across the entirety of the gantry. So we have 12 deck slots here. There's going to be 4 additional deck slots that are available with this configuration. The gripper has the ability to access all of these 16 deck slots, while the pipette has the ability to reach the 12 that are to the left of the instrument. Also, on the front of the instrument is the waste chute. This is going to be utilized throughout the protocol for the gripper to dispose of, in this protocol, it will be tip racks and their lids, but it can also be used to throw away plates or tips or kind of whatever you would like to in the protocol. Moving on, the kind of newest feature that we are debuting here are going to be these OpenTrons Flex stackers here. These are modular, so you can have from 1 to 4 stackers locating on your deck. Here we only have 2 of them, but you can scale up just as easily to have 4. They only take up about another foot of deck real estate, but they do have the ability to greatly scale your capacity. So each stacker can house up to 7 additional tip racks, with 6 going into the actual stacker module itself and 1 being able to sit in its kind of accessory position, and can also store 48 additional PCR plates or 6 additional deep well plates. So in a high throughput setting, such as 96 samples of library prep, including library prep protocols that are more tip intensive, such as ones with enrichment, the stackers can be used to add additional tips and labware to greatly extend your walkaway time. In this setting, once the tips in a particular box are used, the gripper can pick up the empty tip rack, or the pipette tips can be returned to that tip rack, and then the gripper can throw all of it away into the trash chute. The stacker can then move a new tip rack onto the side of the deck in a gripper accessible position, and the gripper picks it up and moves it to where the pipette can actually access this in that now freed up position. Also on this high throughput workstation are a variety of our other modules. There can come with up to 3 or 4 on deck modules. We highly recommend that you have our thermal cycler, which is automatic opening and closing, the new gripper compatible auto sealing lids, which fit on top of labware that can sit within the thermocycler. And this allows you to have multiple PCR cycles run throughout your protocols without the need to do any sort of sterility cleanup, and without having any risks of any evaporation. It also means that you don't need to integrate another module, such as an on-deck sealer and peeler. Instead, have kind of a much simpler solution. The workstation also includes a magnetic block. This is kind of an industry standard 96 well magnet plate, temperature module, which goes from 4 to 95 degrees C, and an optional heater shaker, which goes from ambient to 95 degrees C and can shake from 200 to 3000 RPM. With these, you can truly and fully automate your NGS library prep in a real walkaway fashion. And then as a quick note, we do recommend all of these modules, but the workstation is totally modular. So that means that you're not tied to this robot doing, one specific function or being in one specific kind of configuration. So if you are needing to kind of either scale up in your NGS library prep or kind of move away from it in any way, you have the ability to kind of really quickly, with just a couple of screws in most cases, change out any of the pipettes or modules. And then finally, we also have our on-deck display touchscreen, which allows you to easily store, pull up, and run protocols. It also guides you through any deck setups and automated calibrations, and allows you to change parameters on your protocol from run to run. We will walk through this in greater detail in a minute here as we start this. But most importantly, the touchscreen provides a lot of prompts and a lot of guidance. So it really does make the steps that you go through before the robot starts to run really straightforward and kind of much more approachable for anyone in the lab to carry out. This is kind of independent of whether they have a lot of information and experience with automation or OpenTrons products overall. So I'm going to go ahead and start this protocol here. So I've already gone through and make sure that all of my instruments and my deck hardware align with this protocol. I've also already made sure that all of my labware position checks are in place. And I've already actually chosen my runtime parameters here. So we are going to be running this as kind of a dry run. So throughout the protocol, the thermal cycler will kind of just open and close instead of doing a whole run through. But this does allow you to kind of really change and optimize how protocols work using runtime parameters. Okay, perfect. So now that I've kicked off this protocol, just a little bit more info about what the robot will be doing, and then I'll kick it over to Matt in a minute here to talk more specifically about the science that is actually occurring. In this protocol, the stackers are going to just be using tips, and they're only going to be coming in kind of bringing to the deck fashion. The stackers have the ability, as I had mentioned previously, to house either tips or different types of labware as well. And so they not only have the ability to bring labware to the deck, but also to store that labware. So if you have multiple stackers, up to 4 in a protocol, you have the ability to bring plates out and then also store them into a different stacker. You have the same ability with different tip racks. So if you wanted to save those for future runs, you have the ability to just place them back into a stacker and continue running in that way. Throughout the protocol, you have the ability to do things such as pause the stacker and empty it or refill it. So as I mentioned, you can fit 6 tip racks into the stacker and 1 in this accessory position, giving you 7 extra tip racks. If you needed more than that, you could either scale up to have multiple stackers, or you can inject pause steps into your protocol that will allow the stacker to kind of replenish that overall amount of tips that are within it. The stackers also kind of work in the same way as the rest of our robot. They'll sit on the deck with a kind of an updated side window. So if you have a HEPA UV module, you can still turn the UV on and not worry about any leakage or anything like that. And they'll also come with sensors. So if you open the doors during a run, it'll pause in the same safety protocols that we have throughout the entirety of the robot. Just a minute ago, you also saw kind of our new labware, which is the auto sealing lids, which was just placed on top of our OpenTron's Tough PCR plate in the OpenTron's thermocycler. So the thermocycler is just going to open here in a minute, and you can see that afterwards, we'll use the same gripper to then throw these this plate away. We do recommend that these are only for single use, but they do have really low evaporation rate and guarantee sterility if you are running multiple cycles throughout your protocol. One last comment on the stackers is that they will only be able to be loaded with a single kind of labware per run. So if you have one loaded with 200 microliter tips, that is what is going to come out of that for the entirety of it. So in this protocol, we have one that's loaded with 200 microliter tips and one that's loaded with 50 microliter tips. This is just for ease of use and also kind of ease of loading throughout the protocol. Okay, perfect. I think at this point that I can probably kick it over to Matt to kind of describe in greater detail what this protocol is actually doing on the robot. And then once these tip racks are used, we can also see the stackers in action. Matt?
Thank you, Nick.
Oh, here, sir.
Hi, I'm Matt Nakano, Senior NGS Scientist here at OpenTrons. So my team develops and tests automated protocols for many popular NGS kits. We carry out a lot of library prep, QC, and sequencing to make sure the protocol is really optimized. So I'm here today to talk about sort of what's being run on the robot. I was there at SLIS. You might have seen our poster. So what I did previously at OpenTrons was develop library prep protocols. I started off with something fairly straightforward, the IDT XGen MC version of library prep. So this is taking mechanically sheared DNA, adding on the end prep, the adapters, and going through amplification. So I would do that for, let's say, our target client would be a small clinical lab that is receiving maybe small batches of samples consistently throughout the week, doing small batches of like 24 or 16, maybe even 48. So the idea of automating that would be to have the same protocol being run consistently and completely walk away. So we want to automate the steps and the entire workflow. So moving on from 24 samples, scaling up to 96, that was like the next big challenge. And to do that, you could have done it with multi-channels. We've done tests like that of having a full 96 well plate and using the P50 and the P200 multi-channels, doing it column-wise. But I would have concerns there if you're doing a full 96 plate that the first column and the last column would have some variation because the time it takes for a single multi-channel to go across the entire plate for distributing reagents or something. So the ideal here when I started automating for the 96 channel is for everything to be a single pipette doing it simultaneously for all 96 wells to minimize any variation across the plate. And my goals was speed, so I only wanted one pipetting per step, and also a walk away. I didn't want to have to do any pauses in order to reset tipwares or take it off to a separate thermocycler. So this is a adaptation of the 24 sample IDT XGen MC, now scaled up to 96. And to do that, that has its own challenges. So the reagents now are, instead of a single column, they're distributed into a single plate so that the whole 96 channel can pick up the entire reagent amount and distribute it to the plate. And to do that, we need a lot more deck space because all reagents can't be combined onto a single plate. They have to be sort of plated out individually. And so that takes up a lot more of the deck real estate. So you have your ampere beads, you have your ethanol, you have all your reagents here sort of packed onto the temp deck, kept cool. And so now having the stackers, we're able to put all those tips off the deck and bring them onto the deck only when they're asked for. So if you see right here, it just distributed at 200. It's going to remove the lid. And then it's going to position the tip rack on the tip rack adapters. Picking up 96 tips all at once takes a lot of force. So that's why we have it specifically strengthened tip rack adapters. Then we'll go pick up the tips, go on to the next step, and then replace the tips and then toss it into the trash. So again, this is IDT's XGen MC. So it's simply the first step is doing some N prep, then is doing the adapter ligation, and then it's going through the PCR amplification. So each of those reaction steps will go into the thermocycler for the actual incubation. And that's where we have at least 3 separate disposable lids that go on the plate to keep it sterile from step to step. Yeah, so that's going to continue going on. I do have some data from the validation runs we did that we presented at SLAS, so I can share some slides with you. Yeah, if you were at SLAS, we had some data to present, sort of demonstrating how these tools sort of enable us to do a full 96 sample run. Next slide. Here is a bird's eye view of the deck layout that we were using. You see in orange is the disposable plate lids. The sample plate starts off in the thermocycler. The reagents are actually plated onto a 384 deep well plate that's on the temp module to keep cold. So we're keeping cold the N prep mix and ligation mix and the amplification mix. And you can see the deep wells on the deck themselves have enough space for the volumes for all the ethanols for the 2 separate cleanups, the beads, and 1 for liquid waste. And then off to the side, you see our flex stackers that will be the ones distributing the tip racks into those positions. Next slide. Here's just a little bit of sample data for the red one, we only did 1 column just to save on reagents. This was simply, the IDT, the XGen MC. MC is like the mechanically sheared DNA. Again, this was sheared before the protocol using a Covaris sonicator. So this was using just lambda DNA just for testing purposes. We would shear it and then go through the library prep. And as you see, This is a slide taken from the poster that we were sharing. Here is sort of the size traces showing the uniformity of all the fragments. And you can see a comparison between these 12 samples. And I just did 4 samples manually with Lambda, seeing the difference between the CV, the overall yield output, and the CV of the final fragments. Next slide. Here's the second experiment we kind of ran. Since we're doing 96 samples all at once on the single plate, even though we have our disposable plate lids, we want to make certain that there's no cross-contamination. So in this one, we did a row of 12 samples checkerboarded with another row of 12 negative controls, just water. So we wanted to see that there was no crossover between samples, no fragments would appear or get amplified. And here in the gel trace, we only have like 5 samples and 5 samples of negative, sort of demonstrating that nothing really carried over, got amplified. At the bottom, this was human genomic samples. This is the twist protocol compared to the IDT XGen MC. This one has the fragmentation taking place on deck. So the first step is the fragmentation, then you go through the end prep, and the rest, the adapter ligation and the amplification. So this is putting a few more enzymatic steps on the robot itself. And here, this is showing some of the sequencing data generated off the 2 separate libraries they ran. Basically, we just want to demonstrate that it produces a nice clean library that sequences well. You get a very high passing filter, your Q30 is really good, and yeah, it all maps back to either the Lambda or the human genomic. Yeah, those are the slides kind of that I had prepared to show. This guy's going to continue working. I guess I'll call back Nick.
Perfect. Thank you so much, Matt. So yeah, as you can see, protocol over here is still running. So we just brought out another couple of tip racks here, threw away another tip rack lid, and we'll move that over and kind of keep running this protocol. The entirety of this protocol would keep running. We've only put a couple of tip racks into here just for kind of demo sake. But overall, as you can see, right, this is having 2 stackers, just how many more tips and how much more capacity you can run kind of, in a full walkaway fashion. And you still have the ability to add 2 more stackers to this protocol overall. So I think that we can probably kick it over to Q&A. Fran, I think that you are up to read them off.
Hi, everyone. Yeah, so if you want to ask a question, please go ahead. So there's a Q&A box at the bottom of your screen. If you just click on there, you can just type your question in. And we have plenty of time to answer all your questions. So whatever is on your mind, please go ahead. So we had a couple of questions come in while you guys were talking, so I will start with those. But yeah, please feel free, everyone on the call, to start adding questions. So the first question is for you, Matt. You said something when you were describing the reagent layout about reagents being kept in the 384 well plate. Do you think you could just describe that in a bit more detail?
Yeah, so in this particular situation, since I have 96 channels operating simultaneously, I didn't want to put reagents into a single column so that I would have to use the single column pickup. So even though it can do that, what I wanted to do is to save on time to do a single reagent pickup. So I have the reagents sort of already dispensed into wells on a 384 well deep well plate. So this is where everything is in its correct position. So the wells that would be in the reagents for A1 are in A1. Sort of everything's positioned already in the deep well plate. The other thing is I have maybe like 3 or 4 different reagent mixes, and I want to keep them all cold or all at 4 degrees. So able to put all 4 of them on a single plate on the temp module just to keep them cool. So I don't have to do pauses. I don't have to swap anything out or like pause it, run to my fridge and put something on there.
Okay, excellent. So you had 4 different 3 or 4 different reagents on that plate.
Yeah. And in this particular case, I believe it's a Grenier 200 microliter deep well plate in which it's only recommended for the P50 because the P50 could reach to the bottom of that plate because it is more narrow and deeper because it's a 384 well.
Okay, very cool. Okay, and then just another question. It's actually still relevant. I'm going to answer this question because it's still relevant to what we just talked about in terms of the reagent plating. So you mentioned the reagent plating being sort of all the reagents in one deep well plate and then on one temperature block. Is it possible to use more than one temperature block?
Yeah, it is totally possible to have more than one temperature module. I believe the current suggestion is it's only on the left-hand side of the robot, so only in column 1. So right now in this setup, column 1 is taken up, half of it is taken up by the thermocycler, but position C1 and D1 could both take temp modules.
Okay, great. And actually just, so another question actually still related to this reagent plate, which is great. So how did you fill that 384 deep well reagent plate?
In my particular case, I have a separate OT2 that I do reagent setup. So that is where you put maybe 2 parts prep for 1 part actually running. So I take my time to actually aliquot the reagents into the 384 well plate and run it here. You could have a separate protocol on the Flex itself using the same labware and everything to do a reagent setup. But I just did it myself on OT2 separately. But yeah, a little more time invested in actually setting up a reagent plate rather than just taking stuff directly out of the kit in order to get a smooth, fast run on the actual library prep on the actual robot.
Yeah. Great. Great. Thank you so much. Okay, so let's change tack a little bit and go into some other questions. Okay, so this question here is about what protocols can this platform cover? Can it also do nanopore sequencing? So presumably that's like Oxford nanopore long read sequencing.
I guess that's a question for me. I have done nanopore library prep, not particularly with the 96 channel edge, but with multi-channels. What we were doing was rapid barcode and the genomic ligation. So the throughput for there, at least on the projects I was working on, the maximum would be like 24 samples. So in that case, we had plenty of deck space and the multi-channels performed fine. What you can see out there is a 3D printed STL file for an actual adapter that could put a nanopore on the robot itself. And I have done a small little protocol trick with a single channel to actually load the nanopore, the MinION, on the robot itself. But yeah, we have worked on nanopore protocols.
Yeah. And a good resource is to have a look in our protocol library, which is library.opentrons.com, and that houses all of our publicly available protocols. So you'll see there's lots of library prep protocols on there, and we're adding to them to that resource all the time. We also have a internal field applications team who will, if we haven't developed a protocol for it, will develop a protocol for you. So as long as we can do it, we can do it. You know, we will build it for you. So, okay, moving on. So this is similarly around kind of protocol adaptability. How easy and maybe this is a Nick question. How easy and convenient is it to change the code for tweaking operation procedures? And so maybe you could talk about kind of day-to-day operations versus sort of more sophisticated changes.
Yeah, absolutely. So I think starting to answer that question, for kind of an end user, right, who is going to be operating this robot, they totally have the opportunity to have runtime parameters set up, which can be controlled directly through the touchscreen, which can have things that are kind of those minor tweaks, right? So whether that's how close to the well bottom do you want to be aspirating from? How much volume do you want to be mixing with, right? Even down to like flow rates for those kind of specific optimizations, as well as things like having the ability to have it written out as a dry run. So that's what we just had, right? So we can go through the whole protocol, but not have to wait around for the thermocycler and to work through all of its actual cycling steps. To take another step back from that, we do have kind of two separate authorship tools that we fully support. One of them is going to be our Python API, and this is like a really robust kind of coding solution, which allows you to kind of have a ton of versatility in how you are performing both your application and any optimizations thereafter. It does require the understanding of how to code. If you do go through some of our kind of internal tools, whether that is a protocol from the protocol library or having a protocol written out by FAS, they have the know-how to set up these protocols with the most kind of high value runtime parameters available to you, so you have the ability to optimize off of those. And then we also have our kind of drag and drop GUI tool, which is called Protocol Designer. And so this offers you kind of a really easy tool to create protocols and to go back in and edit those without the need to understand code.
Okay. Thank you very much. Okay, so this is another really good question. How do you minimize individual well variation? I have sometimes seen that library yields can end up varying over 50% with similar DNA input. So I'm not sure if this person is referring to an automated protocol or a manual protocol where they have up to 50% variation. So maybe you could talk about the kind of variation that we typically measure, and then how to minimize that.
Matt, I think that's you.
Yeah. So when we do our library prep, we do our QC, we check it with just a quantitative picogreen, and we also do tape station, and we do qPCR sometimes just to check the variation across. Granted, these are like validation tests. We often start with a very uniform sample, and we know out in the field, once you actually get patient samples, there could be a lot of variability. So what we try to do is minimize sort of any of the pipetting steps. So that's why switching up the 96 channel is using the 96 channel head rather than using a multi-channel and processing each column individually, because you could get variation introduced from the first column to the last column. What I believe they were mentioning at the very beginning is, like, once you get to a point where you want to normalize a whole bunch of samples at once, it's actually incorporating a normalization procedure into your library prep protocol. So I believe with IDT, they have like the normalize module, which is automation friendly. It incorporates an enzymatic step that will sort of normalize the samples across the board that you could put within your library prep protocols. When you're finished, you have something that you can just immediately pull and throw on a sequencer. Hopefully that answers the question.
Yeah, excellent. Thank you very much, guys. Okay, so that was all the questions that we had in the Q&A box. I will just leave it open for another 30 seconds in case someone is frantically typing their question and has something else that they wanted to ask. Okay, it doesn't look like there are any more coming in. Just final call, putting it out there. Okay, I think we're clear. So that's it for Q&A.
Great. Thank you so much. So I think at that point we can wrap it up here. Thank you, Matt, so much for coming on and for answering our questions. I hope everyone is really excited about the stackers. We're really excited to be able to kind of put this solution out there and really have kind of a game-changing amount of throughput in a really kind of like small kind of bench space area at kind of an unbelievable price point as well. So very excited to be bringing this solution to everyone, and clearly it should help with increasing that walkaway time. Perfect. Thank you so much.